Good day, this is Professor Will, CBMD PhD. Today's Thursday, July 8, 2021. It is 6:29 and 51 seconds, p.m. Eastern Standard Time. Today's research, lung cancer. So let's hop to it here. Potential marker for success of immunotherapy in the treatment of lung cancer. Date July 8, 2021. Source, Medical University of Vienna. Lung cancer has the highest mortality rate of all cancers, and treatment options are extremely limited, especially for patients with oncogenic mutations in the KRAS gene. And some patients respond very well to treatment with immune checkpoint inhibitors, while it is completely ineffective in others. A research group identified a potential marker for the success of immunotherapy in lung cancer patients and explained the underlying molecular processes. KRAS, it is a monomeric gene protein that plays a key role in the growth of malignant tumors. KRAS mutated lung carcinomas frequently occur in chronically inflamed lungs, particularly in heavy smokers. The research group has now shown that the expression of the highly anti-inflammatory protein, A20, formed in the body itself, is often very low in these malignant cells, and that there is a direct correlation between a patient's life expectancy and the expression of this protein. Both in humans and in the animal model, the loss of A20 leads to downgraded immune surveillance of cancer cells. Cancer cells with low levels of A20 are able to escape detection by the immune system. This results in significantly faster tumor growth. The research team discovered that it is primarily an enhanced sensitivity of the cancer cells to the immunomodulatory cytokine interferon gamma that is responsible for this. Moreover, tumor cells with downregulated A20 responded particularly well to immune checkpoint inhibitors, in the same way as patients suffering from melanoma, skin cancer, with a similar gene expression structure. In A20, we have discovered a previously unknown tumor suppressor in lung cancer, the loss of which is an immune checkpoint, contributes to the development of this malignant disease. And since patients with low A20 expression have few tumor-fighting immune cells, and so on, in the advanced stage express little of the important immune checkpoint molecule, PD-L1, these patients could be excluded from immunotherapies directed against PD-L1. Indeed, the strength of expression of this molecule is currently regarded as an aid for deciding whether or not they should be treated with immune checkpoint inhibitors. Based on our results and the data available from melanoma patients, we are convinced that we have identified a group of lung cancer patients who would really benefit from this immunotherapy. Exclusion from such treatment would significantly reduce the survival rate of such patients. In a further study, the researchers want to find out whether it is possible to manipulate the expression of A20 in the cancer cells in order to intensify the effect of immunotherapies. However, smoking is still the most easily avoided risk factor for lung cancer. We must therefore support laws to protect the general public from inhaling harmful smoke, while at the same time appealing to people's personal responsibility to refrain from smoking altogether. It is nevertheless important to continue to investigate new therapeutic approaches to improve the quality of life and chances of survival of those affected. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.